tell you about some tricks if you want to get some of the substances into the brain. So what I will talk about here has to do with a very special method where we are able to look into the brain tissue using a microscope. If you have a fluorescent molecule in the tissue, this type of microscope can look into living brains. It can even look into awake living animals that move. That is shown in this movie, if I could get it to work, but unfortunately I don't know about Mac computers. But if I press the button here, you can see this small mouse actually walking on an airlifted horizontal platform, exploring the microenvironment consisting of small pieces of wood. And while it's doing that, we can actually look into the brain and examine the blood-brain barrier. But just a few words about the barrier. The barrier consists of a lot of components. First of all, we have, and this is what most people understand about the barrier, the tight junctions between endothelial cells. People think that that is the barrier and nothing else. Of course, this is a very important part of the barrier, but there are many other elements. We have outside the blood vessel, we have pericytes that are small vascular smooth muscle cells. These pericytes make a basement membrane. On top of the pericytes, but also on top of the endothelial cell, we have astrocytes, especially specialized structures called astrocyte end feet that also make a basement membrane that enclose the capillaries. We have this basement membrane now depicted to the left as a purple structure, which is a very dense structure and full up of proteins. We have on the inside of the vessel, we have a dense layer of glucose molecules called the glycocalyx that consist of polymers of di and and unisaccharides. And we have also, on top of that, we have the axon terminals sometimes getting to the blood vessels, but most of the time they actually end up on the astrocytes. So in addition to the tight junctions that I talked about first, there's a number of elements that has to do with structure. On top of that, we have very powerful systems that actually depress vesicular transport. And vesicular transport is turning out to be more and more the way that drug molecules are getting across the barrier. In the arterioles, we have a molecule called sphingosine 1-phosphate that suppresses vesicular transport in arteriolar endothelial cells, and along the capillary endothelial cells and the endothelial cells in arterioles, we have a molecule called MFSD2A that suppress another type of vesicular transport. And into your brain, you have a molecule like aspirin that enters quite easily. But most other molecules will only get into your brain by transport. That means carrier-mediated transport or vesicular transport. So nothing gets in easily. So therefore, it's quite difficult to treat brain disorders. I will try to, you can have here a summary of the different barriers, but I'll just show you how we have looked into the barrier properties of the glycocalyx in a study that we published some years ago. So that we think have mainly to do with diffusion between two endothelial cells, but whether this is correct or not is at the moment difficult to say because the vesicular transport that are outlined in the remaining part of the slide, I think that is by far most important. So the brain endothelium constitutes the blood-brain barrier, but the vesicular transport is equally important. So we do a two-photon microscopy in these mice, and the mice have a number of fluorescent molecules. We have a craniotomy, and then we can look into the brain by this interesting microscope. And the first type of experiment we carried out when we were looking into the barrier properties of the blood vessels was to look at the inside of capillaries. And what you can see here is that you can fill up the lumen of a capillary. Doesn't really work, the color here. I'll try to use this. But you can see that you can fill out the inside of the blood vessel with a fluorescent dye. And if you can determine the center of the blood vessel, you can calculate the diffusion and the distribution of fluorescence from the center out to the side. If you then give a fluorescent dye with a small molecular weight, it will equally distribute along the blood vessel. But the higher up you get in molecular weight, the more difficult the molecule will have in getting out to the sides. That I will show in the following slides. And a main obstacle for small molecules, but mainly for big molecules, is the glycocalyx, which is the glucose molecule at the inside of the blood vessel. It also exists on all nerve cells, and in fact in all cells in the body. On bacteria, on viruses, you have glycocalyx. So the glycocalyx is a molecule which is rather negatively charged, but is present on a number of cells. It consists of a lot of different glucose molecules that are here, kind of there. Some guys vertically oriented, there are some molecules that are more making a network, but they are, to a large extent, very impermeable to large molecules. So we can put the fluorescent dye in plasma here, and we can work with an empty vessel where there's no red blood cells, and we can work with a filled vessel, and then what you see as a corona around the red cell is in fact the glycocalyx. Then we can calculate how much of an injected molecule that actually gets into the glycocalyx, and we can see here on the top part of the slide. You can see here that if you take a small molecule like fluorescent, it will penetrate completely into the glycocalyx. But you take a bigger molecule, it will only go in to some extent. And we have identified something we call the glycocalyx partition coefficient. So the bigger the molecule gets, the less susceptibility the molecule will have to enter the glycocalyx. So the glycocalyx on the inside of the blood vessel is a barrier of big molecules, and we can look at that for several different molecules. For the small molecules, it's no barrier, but the higher you go in molecular weight, the more of a barrier it is, and the more of these big molecules will be displaced into plasma and will never reach the arterial or capillary endothelial cell wall. So it will never get to any receptor, it will never get taken up. So you have to take that into account if you want to give the mouse or the man, the humans, big molecules like monoclonal antibodies. We can also see that if we label astrocytes outside the blood vessels and we look at the diffusion coefficient in the outside, the blood vessel, and look at the extravascular components, then also the astrocytes are and the astrocyte end feet, between which there are only narrow slits, they also constitute a barrier for diffusion of smaller molecules, but even more so of big molecules. So the barrier that we have to get around is the endothelium with the blood and the glycocalyx. Then if you get through the endothelium, you have to get along the basement membrane, the periside, and the basement membrane on the other side. But if the molecule is lucky enough to get across that barrier, it also have to get through the end feet. So we think that there are three barriers: the glycocalyx on the inside, the endothelium, and the astrocyte end feet. This is how we have defined it. So there's a lot of ways not to get across. But of course, there's also some ways to get across. First, I will talk about a way called absorptive-mediated transcytosis, which is looked at as a kind of a non-specific transport, but it has to do with transport of proteins. It might be of interest to some of you guys doing peptides and biologics, because that's one way of getting stuff across, that is to use absorptive-mediated transcytosis. So the way this works is that you have a molecule that's a bit positively charged. It will bind to the negatively charged glycocalyx on a cavevolae, a small endocytic peak in an endothelial cell membrane. It will then become a vesicle that get transported across the membrane. It turns out that this process that looks like non-specific is highly regulated. And the molecule that regulates it in brain is called sphingosine 1-phosphate, and it binds to a receptor called sphingosine 1-phosphate receptor. And we can make a mouse that is knocked out for this molecule here, or has a lower level of molecule. And what happens when you lower this concentration of the molecule and the binding to the receptor is that you have an increased vesicular transport in a lot of tissues. We found it first in the lungs, and then we thought about the brains. And also it seems that this molecule has to do with suppression of the paracellular transport by making tight junctions and adherent junctions more stable. And it turns out that also if you give an agonist of the receptor, that you can actually rescue the blood-brain barrier. So what you can see here is kind of an in vivo image. It's showing small vesicles inside an endothelial cell, and they are fluorescently labeled, and you can see how they move. Some people think that these vesicles are actually quite, they're kind of just not doing anything, but they're actually moving all the time. And what we can see is that here in arteriolar endothelial cell, if we take away this molecule, sphingosine 1-phosphate, which might sound exotic, but is kind of a major molecule in the barrier, you actually can get an increase of transcytosis. So here we have a healthy brain on top. You can see here, if you inject fluorescent albumin, nothing gets into the brain. If you take away sphingosine 1-phosphate, then you actually take away the suppression of the vesicular transport. You get albumin accumulation outside the blood vessel. And you give an S1P, sphingosine 1-phosphate agonist a few hours ahead, you can actually rescue the brain. So one of the ways that you guys might want to get your biologics into the brain is by using absorptive-mediated transcytosis, which mainly works at the level of arteriolar endothelial cells, but there are also a lot of vessels involved in many brain diseases, including small vessel diseases. You can see here clearly the difference between an arteriole and a venule, that you get these molecules, they actually get into vesicles in arterioles, but not in venules, so it's kind of located more or less specifically to arterioles. And we can make different graphs of this. But the main message is that there is a specialized vesicular transport, mainly localized to arteriolar endothelial cells. You can go into another type of vesicular transport called receptor-mediated transcytosis, where you can link your therapeutic molecule to a ligand that can bind a receptor on the surface of endothelial cells. Mostly, we use transferrin receptors as the receptor that we link our substances to, because it's highly enriched and expressed on the surface of brain endothelial cells, and it's actually the kind of the benchmark for all type of, if you want to get anything into the brain, you have to compare it to receptor-mediated transcytosis mediated by transferrin receptors. What we have used to test this substance is nanoparticles that are covered with antibodies against the transferrin receptor. They have fluorescent tags on, and they also have some polyethylene glycol, some they are pegylated, so they stay in solution. And the way it works is that they bind to the surface of the endothelial cell, it gets endocytosed, and then some of it will go to lysosomes, but other parts will get into the brain. By vesicular transport, And this is very, in normal individuals, it's suppressed, but you can get something across if on a lucky day. So it works the way you inject the nanoparticle. You can see how it circulates in the blood. This is all from awake living animals. You can reconstruct the brain circulation by making serial sections, serial images like this. And then what you can see is that these nanoparticles are quite stable in plasma. So you have the molecule there for a long time, and if it's there for a long time, it also has a long time where it can bind to the surface of endothelial cells, and that will increase the probability that the molecule will get across. You can see here how it binds to the surface of the endothelial cells. The red color is the binding of the surface. You can see it up here, where you have the red is the nanoparticle, how it binds to these vesicles. And here you can see how actually the nanoparticle gets into the brain. Oops. You can see here how they actually move on the surface of the brain endothelial cell. And here you can see one nanoparticle getting to the surface of the brain endothelial cell and actually getting into brain's extracellular space. And they do so mainly at the level of postcapillary venules. And this, we think, is because that there's a lot of space about perivascular, postcapillary venules. They have a large perivascular space, so there's a lot of room for the nanoparticles to get through. Boy, yeah, I'm in a hurry now. I can see my chairman is actually standing up, or the next speaker. What, 2 minutes? Yeah. You can see how one of the nanoparticles, how they actually move in the brain extracellular space. And if you have a transferrin receptor kind of functionalized nanoparticle, it will also go and bind to the surface of neurons, because the neurons also take up a lot of iron, because they need it for the mitochondria and other enzymes. So it's a kind of a two-in-one type of advantage for having transferrin receptor targeting antibodies. First, they bind to the endothelial cells. Second, they bind to the surface of the neurons. So they will get in and could target, for example, tau or maybe elements of the Lewy body or whatever. Well, maybe I should go to the last slide here. So in the arteriolar end of the microcirculation, you control the blood flow through the capillary tree. And this is also where you have absorption-mediated transcytosis. Along the capillary, you have kind of an exodus of glucose and oxygen and amino acids and other small molecules. And then at the venous end, you can get receptor-mediated transcytosis of nanoparticles. And this is also where immune cells get out. For example, if you have multiple sclerosis. So the multiple sclerosis disease is not like immune cells getting in all over. They're only getting in postcapillary venules. So I think we need to understand better how the microcirculation works in order to affect it. So with this, I will thank my colleagues, Christoph, Alec, Nikolai, and Mette, who's done the work, and you for your attention. Thank you very much.